DNA sequencing is the process of determining the exact order of the chemical building blocks that make up DNA. These building blocks, called nucleotides, are represented by four letters: A, T, C, and G. The sequence of these letters forms the instructions for building and operating living organisms. DNA sequencing is like reading the text of life's instruction manual, letter by letter, word by word. Understanding the sequence of DNA helps scientists learn how genes work, how organisms are related, what causes diseases, and how life evolves. Every organism has its own unique DNA sequence, whether it's a human, a plant, a bacteria, or a virus. Knowing this sequence allows us to answer important questions, such as, which gene causes a certain disease? How closely related are two species? Is a person at risk of a hereditary illness? How did a virus change over time? This information is essential for medicine, biology, agriculture, forensics, and evolutionary research. Think of DNA as a massive book written using only four letters: A, T, C, and G. DNA sequencing is the process of reading the entire book, page by page. If one letter is out of place in a critical word, it could change the meaning, just like a mutation in DNA may cause a disorder. Sequencing helps us identify those changes. Although different technologies exist, most sequencing methods follow three main steps. Break DNA into small pieces. Determine the order of letters in each piece. Chemical or optical techniques detect which nucleotide is present at each position. Reassemble the pieces in the correct order. Powerful computers stitch the short sequences back together to reconstruct the full DNA sequence. The final result is a digital file, a long string of letters representing the DNA. The first human genome, completed in 2003, took 13 years and cost $3 billion. Today, the same task can take hours and cost under $1,000. This dramatic improvement has transformed biology and medicine. Doctors can identify genetic disorders by comparing a patient's DNA to typical DNA sequences. Diseases like cystic fibrosis, muscular dystrophy, or hereditary cancers can be detected early. Sequencing helps doctors tailor treatments to individuals. Cancer cells have mutations. Sequencing tumors helps doctors choose the best therapy and track how cancer changes over time. DNA sequencing is used to identify suspects, match biological samples, and solve crimes. Sequencing tells us how humans migrated across the globe, how species evolved, and how individuals are related. Scientists develop crops resistant to disease, drought, or pests by understanding plant genomes. Sequencing helps monitor outbreaks, such as identifying COVID-19 variants. DNA sequencing is powerful, but challenges exist. Large amounts of data must be stored and analyzed. Errors can occur during reading or assembly. Some genomes are very repetitive, making them tricky to piece together. Advanced algorithms and high-quality lab techniques help overcome these issues. As sequencing becomes cheaper and more common, questions arise. Who owns your genetic data? Should insurance companies have access to it? Can employers use genetic information unfairly? Should we know everything about our genetic risks? These issues require careful policies to protect privacy and prevent discrimination. DNA sequencing is the process of reading the genetic code that shapes every living organism. It has revolutionized science and medicine by allowing us to understand how genes influence health, disease, and evolution. As technology improves, sequencing becomes faster, cheaper, and more widely available, opening new possibilities for personalized healthcare, better agriculture, and deeper biological knowledge. Put simply, DNA sequencing lets us read the instruction manual of life, revealing how living things work, where they came from, and how we can help them thrive.